Good afternoon. So, the topic for today's discussion is regulation of enzyme activity, and it's coming under Biochemistry Competency 2.3.3. So, myself is Dr. Shyam Krishnan. I am Assistant Professor in the Department of Biochemistry, MMC. So, what is the specific learning objective of this particular topic? After this class, the student should be able to explain the various factors which regulate the activities of enzyme. Already enzyme has been covered, enzyme different types, what are the classification systems of enzymes, factors affecting enzyme activities, everything has been covered. So, this will be adding upon to the topics. So, enzymes as we know, these are basically proteinous molecules which are acting as biocatalysts in our body. But there may be some enzymes which are exceptions like ribozymes, which are RNA molecules, and if you are considering how these enzymes are regulated, basically we can divide it into two. So, first portion is the regulation of enzyme quantity, that means the amount of enzymes is regulated here, and this is the regulation of enzyme activity, that means the activity of the enzymes or the quality of the enzymes, how it is acting, it is being altered in this regulatory mechanism. So, we will be dealing with one by one. First, we will be dealing with the regulation of enzyme activity. So, the regulation of enzyme activity or enzyme quantity, it is basically by the control of enzyme synthesis by inducers and repressors. So, again this topic was covered along with the factors affecting enzyme activity. So, I will not be going into much detail of the topic. What is an inducer or what is a repressor? An inducer or repressor, as it is written here, it is a molecule that is acting at the genetic level, which is noticeable after a lag period of hours or days. So, what is the word meaning of that? It means that the inducers or repressors are not directly acting upon the enzyme molecule. It is acting at the genetic level. We know genes are being transcribed into RNA, it is being translated into proteins which are having the enzymatic roles. So, it is acting at a genetic level means it is directly involved in the amount of the protein or amount of the enzyme that is produced. So, what are the examples for that? We just need to know a few examples for each. And inducers, as the word meaning, which increases or stimulates the synthesis of enzyme molecules like insulin inducing glucokinase, the glucocorticoids inducing transaminases. All these you can write as examples for inducers. Whereas repressors, it will decrease the synthesis of enzyme molecules. The repressors molecules, it will go and bind to the genes, and it will be, what it will do is that it will be decreasing the production of these enzyme molecules. So, what are its examples? The commonest example that we can say is that of heme. Heme, as we know, it will be acting as a repressor of its own synthesis. The ALA synthase is one of the initial enzyme involved in the synthesis heme synthetic pathway. So, the final product heme, it will be inhibiting ALA synthase by acting at the genetic level. It will be acting as a repressor, and ultimately it will be decreasing its production. Another type of regulation that is coming under the quantitative regulation is feedback regulation. So, feedback regulation means some pathway end products control their own synthesis. In many instances, it is by feedback inhibition, that means directly acting on the enzyme levels, but that is different from feedback regulation. Feedback inhibition is a type of enzyme inhibition. Feedback regulation is more of a regulatory mechanism, it is a broader term, regulatory mechanism. So, one example that we can say here is HMG-CoA reductase, the rate limiting enzyme of cholesterol synthesis. It is regulated, but cholesterol is not actually acting directly upon this HMG-CoA reductase. It is acting at a different regulatory mechanism. It is having a different regulatory mechanism, which is the feedback regulation. So, regulation in response to dietary cholesterol involves repression of the gene that encodes this enzyme, that is HMG-CoA reductase, by cholesterol or its metabolite. So, the cholesterol or its metabolite. The cholesterol, it is not directly acting upon the HMG-CoA reductase at the protein level, it will be acting upon the genes that are synthesizing HMG-CoA reductase, and it will be ultimately manifest as decreased production in case of increased cholesterol in availability. That is another regulatory mechanism. So, next is how we can control the already produced enzyme. The quantity can be regulated by the controlling the enzyme degradation. So, first pathway that is involved here is ubiquitin proteasome pathway. it is not just for the enzymes. Ubiquitin proteasome pathway is a general mechanism for proteins which is degraded, or the degradatory mechanism for proteins. So, in this pathway how enzymes are regulated is that proteins are degraded by the ubiquitin proteasome pathway. Every protein, not just enzymes, almost all proteins in our body undergo this pathway for degradation. So, the enzymes, there are also mostly proteins, they are mostly undergoing this type of regulatory mechanism or this type of degradatory pathway. So, this is what is a animated video of how this is happening. You can see it is like ubiquitin proteins, ubiquitin tagged proteins are going on to the proteasome. So, this is the intact protein, it is going into the ubiquitin proteasome system, and now you can see the degraded products coming out. So, ubiquitin is acting like a tagging molecule. You can see the violet colored tagging molecule in this animation video. So, if it is tagged with ubiquitin, it will go into this proteasome structure. That is how the mechanism works. So, ultimately the intact protein, it is being degraded into different products, and it will be later cleared from the system. So, here we are not changing the production of enzymes, we are dealing with the regulatory mechanism is basically concentrated upon the degradation. So, degradation is one path of regulation. If there is degradation, there should be some mechanism for stabilizing the enzymes. That means the enzyme activity needed to be continued or pursued. That is stabilization mechanisms. So, some enzyme molecules undergo wear and tear and are finally degraded. That is a usual happening. So, what we want is that we do not want to degrade the enzyme, already produced enzyme, we want to prolong its action. Somehow it has to be stabilized. So, what are the stabilization molecules or stabilization techniques that are possible? So, by preventing such degradation, what we can do is that we can increase the overall enzyme activity, and this process, as explained, it is known as the stabilization of enzymes. So, one example we can commonly say is degradation of tryptophan ferrulase. The enzyme is prevented by tryptophan. And another example that is commonly mentioned is growth hormones acting on phosphofructokinases. This enzyme phosphofructokinase is stabilized by growth hormones. So, any enzymes with SH group, that is the sulfate group, SH group, sulfur containing groups like papain, urease, succinate dehydrogenase, etc. are stabilized by the molecule glutathione. So, all these molecules, what they do is that they help to increase the half-life or stability of these enzymes. So, that was about the regulatory activity. We have discussed about the quantitative regulation of enzymes, starting with the production and degradation part, including the stabilization part. Now, we will directly move on to the regulation of the enzymatic activity, the catalytic activity of the enzymes, how it is regulated. So, first one is allosteric regulation. The word meaning of allosteric, as we know, or you may be familiar with those word meaning, that allo means other site. It is acting at a site other than the active site. So, it is spatially different or distinct from the catalytic site of the enzyme, but it is having some impact on the final action of the enzyme. The commonest model that is described for this allosteric regulation is aspartate transcarbamylase, which is an allosteric enzyme involved in the pyrimidine biosynthesis. This enzyme will be dealing with when pyrimidine metabolism is taken, but just for the time being, just know that it is a allosteric enzyme. So, the end product of this pathway, that is CTP, cytidine triphosphate, this pathway inhibits the enzyme ATCase, that is the aspartate transcarbamylase enzyme is inhibited by its end products CTP or cytidine triphosphate in an allosteric manner. Whereas, it is not just inhibition, the ATP molecule, the purine nucleotide ATP is an activator, allosteric activator of this. That means, regulation involves both activation as well as inhibition. CTP is an inhibitor, ATP is an activator. So, you can see what is happening in allosteric activation and allosteric deactivation or allosteric inhibition. So, in allosteric activation, what happens is that when the regulatory molecule is binding, you can see the shape changes. The shape was like this, it is changed to something that is fitting into the substrate can fit in, so that the reaction takes place. That is, it is welcoming the substrates, the reaction is taking place and it is activated. That is what happens when ATP binds to aspartate transcarbamylase. Whereas you can see here, it is allosteric inhibition. when this regulatory molecule CTP is binding, what happens is that the shape changes so that there is no possibility of the substrate binding. Even if the substrate comes here, the shape is not welcoming the substrate, or substrate is not able to fit into this shape. So ultimately what happens is that there is no product formation. So that is how allosteric molecules work. The binding of this allosteric molecule alters the three-dimensional structure, that is the tertiary and quaternary structure of the protein is altered, so that the active site also having some changes in the shape. So either sometimes it can lead to activation, sometimes it can lead to inactivation or deactivation. That is allosteric regulation. So next is the multiple forms of the same enzyme, that is isoenzymes. So you can see all these are 1 rupee coins. This is a 1 rupee coin maybe made in 2001, this is a 2005 made coin, this is a 2011 made coin. These, we cannot say that these are all the same. These are different coins. These are different, structurally different coins, but they are doing the same function. If you give this coin or this coin, any coin to purchase something that is worth 1 rupees, you will be able to do that. So these are structurally different coins having the same function. Likewise, the term isoenzyme, it denotes that these are enzymes. These are enzymes which are having the same function, but they are structurally, origin wise, even the gene of their origin are different. Only thing is that they are all having the same function. And isoenzymes as such is a separate topic for you, it will be dealt in later. And isoenzymes, the thing that you have to remember is it is having the same function, it may be present in different tissues, it may be originating from different genes. All the physical properties, their electrophoretic properties, their migratory properties, the suppression by chromatography, everything may be different, but functionally they are the same. So how it is helping, or it is acting as a separating medium, or how it is acting as a regulatory molecule is what we are going to discuss. So since they are catalyzing the same reaction but present in different tissues, what happens is that, and because of the structural differences, there will be differences in the Km value and the Vmax of the enzyme. You know what is Km, it is a Michaelis constant, and Vmax is the maximum velocity that is attained by enzyme. That will be different, as well as the regulatory properties will be different. So what happens is that even though, suppose isoenzymes, one example we can say is glucokinase and hexokinase. Glucokinase is present in liver basically and pancreas, whereas hexokinase is present in all tissues. So how their regulatory activity is different is what is written here. I think this topic was already dealt with during carbohydrate metabolism, so there is no much need to delve too much deep into this thing. We will be just using this example to explain the concept of regulation by isoenzymes. So the glucokinase, it is induced by insulin. That means if insulin is produced, there will be increased production of glucokinase. That will be the induction or increased production of glucokinase by insulin, whereas hexokinase is not. That means hexokinase, even if insulin is there, there is no increased production of hexokinase. It is not responding to insulin. So as I have told, isoenzymes, they are expressed in different tissues or organelles. So again, that is a method of regulation or how the enzyme activity is controlled. So the next is covalent modification, which you may be coming across or you might have studied in enzyme factors affecting enzymes, maybe previously also in so many other pathways we have already studied this regulation, how covalent modification is acting as a regulatory mechanism. So reversible covalent modification means the catalytic properties of many enzymes are altered by covalent attachment of a group. That means a covalent bond is formed, and the most common covalent bond formed is phosphoryl group. That means phosphorylation is the most common or the commonest covalent modification that is present in our body. Who is phosphorylating them? Who is the phosphate donor? In almost all reactions in our body where phosphate is added, ATP is the donor, and these are catalyzed, or the addition of this phosphate group is catalyzed by enzymes known as protein kinases. And sometimes phosphate group is added, sometimes a phosphate group is removed, that is known as dephosphorylation. So the dephosphorylation or removal of phosphoryl group by hydrolysis, it is catalyzed by protein phosphatases. So, this example you might have studied during glycogen metabolism. Glycogen synthase, it is active in the dephosphorylated state, whereas glycogen phosphorylase, it is active in the phosphorylated state, and the role of glucagon, insulin, etc. are detailedly described during the carbohydrate metabolism. So, I do not think there is any need for further repeating the same thing which you already know of. So, irreversible covalent modification. So, irreversible covalent modification means basically it is hydrolysis of proenzyme into an active enzyme, or hydrolysis of zymogens into its active form. So, where you have come across these hydrolytic reactions? Mostly you will be aware about the digestive enzymes. So, I think you know about the enzymes chymotrypsin, trypsin, pepsin, etc. You might have studied in +2 classes, or you might have studied during digestion topics, or even in physiology the same topics are covered. So, what happens is that these are not produced as chymotrypsin, as trypsin or pepsin. These are produced in its inactive or pro-enzyme form. So, what happens is that if you want to convert this chymotrypsinogen or trypsinogen in pepsinogen into trypsin, you may have to cleave, or you have to break a small portion from the peptide, and then only the rest of the protein is acting as the active form, or is acting as the active protein enzyme. So, if that cleavage is not happening, what happens is that it will not be activated. So, chymotrypsinogen will be staying there as chymotrypsinogen, trypsinogen will be staying there as trypsinogen. Ultimately, the reaction or the action of the enzymes are not happening. So, this is one mechanism of regulation of the enzyme activity. And one more thing, these digestive enzymes or clotting enzymes, it should be shut down. It's not that suppose coagulation pathway is going, the different clotting factors are activated. They are, most of them are serine proteases enzymes. So, it is going on acting. So, it is not good. You have to have a stop, or you have to have a termination point. So, even that termination point or shutting down is also done by irreversible binding of the specific inhibitory proteins, So, next mechanism of controlling the enzyme activity is compartmentalization. Compartmentalization means this is urea cycle. I am not asking you to learn the urea cycle now. I am not going into the details of the urea cycle. It will be a separate class. But what you should know is that you can see different compartments here. Mitochondria is here. This is cytoplasm. So, one portion of the cycle happens in the mitochondria, other portion it is happening in the cytosol. So, what is the advantage of this? Advantage of compartmentalization is that it is providing an additional opportunity for regulating the enzyme activity. Suppose this molecule has to come out here of the mitochondria into cytoplasm. That transport, you can have a control. You can regulate the transport of this molecule into this, like citrulline out of this mitochondria into the cytoplasm, so that when the cycle is saturated, the transport mechanism can be down regulated, or when it is free to operate, you can increase the product, and there is increased production of ammonia, you may need to increase transport of these molecules and activation of the cycle. So, what happens is that in this regulatory mechanism, when one portion or mitochondria is having one portion of the cycle, cytoplasm is having another portion of cycle, there is a transport mechanism, transporter is there in between. So, what happens is that provides an additional measure or additional opportunity for regulating the enzyme activity. So, that is what is already explained. I am just giving it as some notes. So, the compartmentalization of this pathway, like into mitochondrial pathway and the cytoplasmic pathway, I have explained with the example of urea cycle. Other pathways like heme synthesis, gluconeogenesis, etc. all share this common property, so that an additional measure or additional opportunity for regulation is there because of the shuttling of the intermediates from mitochondrial membrane to outside and vice versa or vice versa. So, till now this is a small topic, regulation of the enzyme activity. Basically, we broadly divided it into enzyme quantity and enzyme activity. So, regulation of the enzyme quantity, we started with these inducers or repressors, and then we discussed about feedback regulation. Then we discussed about the degradation pathways of the enzyme, ubiquitin tagged proteins, how they are, or enzymes, how they are degraded, and we have seen an animated video of what is happening to ubiquitin tagged proteins. And after that, we have discussed about stabilization, how to prolong the action of already formed enzymes, stabilization of enzymes. Then we moved on to regulation of the catalytic activities. So, the different mechanisms that we have discussed for the regulation of the catalytic activity, like we discussed about covalent modification, we discussed about allosteric regulation, we discussed about compartmentalization. Before that, we discussed about the isoenzymes. All these are producing a method of regulating the enzyme activity. So, this portion, enzyme regulation, has to be read as a continuation of the enzyme chapter, the factors affecting enzyme activity, the mechanism of action of enzymes, enzyme specificity. Continuation is regulation of the enzyme activity, and that's all for today. Thank you.